Welcome back friends, welcome back in another video tutorial for microbiology with Shagnik. We are discussing a lot about protein sequencing and assays. This video will going to be a continuation of that video lecture series. So today we will going to talk about the peptide sequencing. So this technique was first discovered by scientist Edman, and according to its name, this technique is also known as Edman degradation. Okay, so he have used a chemical named phenyl isothiocyanate. That is why this chemical is also known as Edman reagent. So what is the actual thing? This phenyl isothiocyanate will going to react with the N-terminal amino acid residue of the peptide. That is what we have also learned in case of our FDNB and dancyl chloride thing. So by reacting with the N-terminal amino group of the peptide chain, it will form a complex. Okay, now this complex can be removed from the rest of the peptide, and then by further analysis of the complex we can understand which kind of amino acid it is. And by using this technique several times, by using sequential removal of the amino acid residue from the N-terminus of a polypeptide, we can find out the entire sequence of the peptide. Okay, so we will going to also talk about the mechanism, how it is done, then it will be more clear to you. So this is the structure of the reagent phenyl isothiocyanate, and this is a peptide. It is its N-terminus, and this is its C-terminus, right? So now first, in alkaline pH, which is pH 9, mild alkaline condition, this phenyl isothiocyanate will going to react with the N-terminus of the peptide, right, and form a complex which is known as PTC complex, or phenyl thiocarbamoyl derivative. Okay, or phenyl thiocarbamoyl peptide derivative. Okay, so now we will going to remove this part. Okay, and for that we will use acidic condition, that is in presence of anhydrous trifluoroacidic acid. Okay, that will give us a complex named thiazolinone derivative. Okay, and as you can see, it is removed from the rest of the peptide bond. Okay, and that is the idea of Edman degradation. Now this thiazolinone derivative will be extracted into an organic solvent, and then by treated with aqueous acid, and we will get a complex which is more stable one, that is phenyl thiohydantoin derivative. Okay, so for remembering that phenyl thiohydantoin derivative structure, you can just remember that the stable form of the thiazolinone derivative, that is phenyl thiohydantoin derivative, is nothing but a interaction between this C double bond O with this NH, and obviously this H will be removed. Okay, if you carefully look the picture, then you can easily understand. This N reacting with that C double bond O, then CHR, NH, and at the last C double bond S. So that is the idea. That's how we have now, by further analysis of this phenyl thiohydantoin derivative, we can find out which kind of amino acid it is, because as you can see, the R group is still there, and by determining or finding out this R group, we can find out which kind of amino acid it is. Then we will going to do the same thing with this peptide sequence and find out which kind of amino acid it is. Then we will repeat in the case of R3 also, and that is how we can find out the entire amino acid sequence of a peptide. Okay, so that's the video of our Edman degradation. I hope this video will be helpful for you. Thank you.